without H. Hi folks, what I'm here to do today is sell. Do not adjust your television. What you are about to witness is something so magnificent that it can only be brought to you by life. And life can only be brought to you by cells.
Hey, this is about cells. That's great, that's what today's show is about. Oh, hold on, this is today's show. Whoa.
Bill Nye the Science Guy.
Growth is a crucial aspect of our life. Be it growing over the year to be able to go on that one ride you always wanted to go, or just growing up to make your own decisions and handle your own responsibilities. In science, the definition of growth is narrowed down to the increase in the number of cells or the increase in the size of cells. Hence, when you get taller an extra inch over the summer, you've grown. Or when a seed develops into a plant, the seed has grown or germinated. Every living organism is capable of growth and is essentially what classifies them to be living things. That's why inanimate objects such as your phone or the chair you sit on are considered to be non-living things. And that's why viruses are not disclosed as living things, as they need a live host in order to replicate. Alongside growth are several other factors that make living organisms, well, alive. This includes movement, respiration, sensitivity, nutrition, excretion, reproduction, and our very own growth. I'm not going to ponder much over the basis of the definitions of some, but let me just say these are the basic criteria for things to be classified as living organisms. But let's come back to our old pal, growth. We know that growth is what living things do, but what we don't specifically know is how they do it. It's not like movie magic that an 8-year-old boy turns into an 8-year-old man. It's science, and it's over time. To be more specific, it's your cells multiplying and dividing into processes, mitosis and meiosis. Mitosis only applies to diploid cells dividing to form more diploid cells. Diploid cells are cells that have pairs of chromosomes, such as normal human cells have 46 chromosomes or 23 pairs of chromosomes. Another type of cell is a haploid cell that have just 23 chromosomes. This is important to consider since only diploid cells can be formed through mitosis and only haploid cells can be formed through meiosis. So far, we've taken a brief overview of the cells involved in mitosis and meiosis, but for the rest of the video, only mitosis will be discussed.
In the course of human events, this is all pretty recent, right?
Yes.
They found nuclei, there's cells around nuclei, animals and plants have cells.
Yes.
Then what happens next?
Well, after a gap of some 30 years or so, people began to see cells dividing.
Cell division is the process that the cell of a plant or an animal goes through to duplicate itself. While scientists had known about the process for decades, German zoologist Walter Flemming was the first to describe it and to publish his findings.
So Walter Fleming's studies showed cell division in the process of mitosis, where the division of a nucleus results in the production of two daughter cells, each with identical sets of chromosomes in the nucleus to the parent cell. This then results in the formation of two genetically identical diploid body cells. But let's back up for a second, because for mitosis to occur, a lot of things have to be in place. For example, the dividing cell must copy each chromosome before it divides. This includes the DNA replicating and more proteins being added to the structure, not forgetting the cell organelles such as the mitochondria also double. Each daughter cell will then be able to receive a copy of each chromosome and each molecule of DNA when the cell divides. Also, the dividing cell must divide in such a way that each daughter cell receives one copy of every chromosome. If it does not do this, both daughter cells will not contain all the genes. Okay, so these requirements must be attained before the start of mitosis. Hence, they occur during the interphase stage of cells in the cell cycle, which looks like this. Remember that for the majority of a cell's lifespan, it will be conducting its intended function during the interphase. And whilst we are on the topic of the interphase, it's worth mentioning that chromosomes can have different names depending on their form. For instance, chromatin is one form in which chromosomes become uncondensed, whereas chromosomes themselves are condensed chromatins in the form of two identical sister chromatids held together by a centromere. After the interphase, however, the first stage of mitosis, prophase, occurs. In prophase, the chromatids become visible, joined at a centromere, and the nuclear envelope breaks down. Then in the metaphase, a structure called the spindle forms. The chromosomes line up at the equator of the spindle, in other words, its middle, and they are attached to the spindle fibers by the centromere. What follows next is the anaphase, in which the spindle fibers shorten and pull the chromatids to the opposite ends or poles of the cell. Then the chromatids separate to become the chromosomes of two daughter cells. Then lastly, in the telophase, two nuclei form at the poles of the cell. The cytoplasm starts to produce two daughter cells, with each daughter cell having an exact copy of each chromosome from the parent cell. By now, mitosis is finished and cytokinesis occurs. The cell membrane splits to form two separate cells, with the cell organelle being shared between the two daughter cells. The applications of mitosis in the human body is vast and operates whenever cells need to be replaced in our bodies. The skin loses thousands of cells every time we touch something, hence a layer of cells beneath the surface is constantly dividing to produce replacements. However, some growth may not be welcome, such as that of cancer cells dividing by mitosis, in which the cells formed are exact copies of the parent cell, including the mutation in the genes that makes the cells divide uncontrollably.
During cell division, the chromosomes undergo dynamic change. They divide into two identical parts, one for each daughter cell. Fleming called this process mitosis.
So that's the whole process of mitosis as he described it in 1881.
Okay, so hopefully now you should be able to describe the importance of mitosis. You should then be able to describe the stages of the cell cycle involving mitosis.